For this last session today, we'll have actually triple talk. So it will be shorter talks from the people from Google Zurich. So we'll start with Eyvind, who will present us life in Z80 at EPFL.
Well, thanks everyone. Also want to take the time to acknowledge that this was work with a lot of people at Google Zurich, and a few of them are here. So Alex and Ettore are here. Where is Alex? Oh, there he is. Both of you, please feel free to just jump in if I'm saying something that you don't agree with. But yeah, it was a big team effort, so not taking credit for everything. And we were asked to talk a little bit about our journey to Alife. Mine really started through Google. So I joined Google through a kind of alternative path, which was this AI residency they were running for a few years, which picked people out of non-PhD tracks or PhDs in other fields to try and do research, basically. And the first part of that process is to find a team. So for the first 2 months, I was just going around talking to a lot of teams in Google Zurich, a lot of them doing very interesting things, kind of very traditional deep learning things. And then the very last day of like team matching, I had emailed Alex a few times, he never replied. But Ettore reached out to me and said, you know, Alex has some time today, why don't we meet up? And they took me to this sort of padded room that had like very thick walls, so soft, so you could do VR experiments in there. But there was no VR, they just sat me down and showed me on a laptop this very first demo of NCAs, which was a butterfly we're getting this case to. And I was really sold at that point, so I declined the other teams I've been talking to and joined them, and this was my first project, kind of working on this very first article we published together, growing NCAs. And then that's a small part of the Alife community, I think. The rest of it I kind of received through osmosis, through Ettore, who was always pitching, we need to find systems that can increase their own complexity. We don't have that, so I've been kind of sold on that as well. And now we're all at this kind of new team in Google called PI, Paradigms of Intelligence. It's technically a sister team to Google Research and DeepMind, looking at a little bit more esoteric research. I was actually founded on this research, so kind of the founder pitch for that team was BFF, pitched by Blaze, who leads the team. But now we do a lot more things, so NCAs kind of got absorbed in there. Yeah, that's kind of my path to Alife. I'll try and, BFF is kind of an odd beast, and I think many of you have read the paper that we published. I'm hoping to kind of recap that paper and hopefully give it a bit more like intuition for how it works in the first few slides, and then also show just mostly empirically some interesting behavior we see in a different version of BFF, which is called ZFF. So I'll start with that. What is BFF? It comes from this language called brainfuck, which is the BF in BFF. I actually don't know what the last F stands for.
Whatever.
Brainfuck was actually a Swiss invention. I think the guy still lives in Switzerland somewhere. You can think of it as a very simple programming language that implements a Turing machine. So it has 2 tapes. It has an instruction tape, which contains a series of these instructions, and it has a data tape which you can read and write to. And kind of like a prototypical Turing machine, it can move this head it has on data tape backwards and forwards with these instructions. Change what's at the data tape at the current head by incrementing or decrementing it. And then you can do 2 interesting things, which is output a byte from the data tape to your screen, or read 1 byte of data from your keyboard into the data tape. And finally, it has a simple looping mechanism, which just checks for a null byte, and if this byte is 0, then it jumps over the loop, otherwise it enters a loop at this head in the data tape. So how do we go from this simple language to what we use for BFF? Actually, sorry, let me give you an example of a BF program. This outputs hello world to the screen. At some point I did kind of walk through it and figure out how it works. But I think it just loops and increments the values until they're the right ones, if I remember correctly.
There are printing instructions here?
Yeah, sorry. So these 2 are the special instructions. You can output 1 byte of 1 byte from the data tape to your screen.
And you output like binary ASCII or
Yeah, exactly. Yeah. So how do we go from this to what we're using for experiments? Change some things. So now instead of having 2 tapes, you only have 1 tape. And this 1 tape contains both the instructions and the data you're using for your program. And this makes it naturally self-modifying. So if an instruction calls to increment a byte on the tape, it might increment its own instruction or another byte on its own tape. And then we do this process of taking tapes from a soup of tapes, which I'll explain in a bit, and pairing them together by concatenating them. And there's a few other modifications we played around with, including kind of randomly stopping them after some time, randomly mutating bytes in the tapes, and you can also choose or not choose to have non-coding bytes present.
Just to clarify, there's 3 tapes you can think about. One is the instruction, one is the input, one is the output. Here, everything is the same tape. So input and output reside in the same place as well as instruction.
That's a good point, sorry, yeah. Well, yes, yeah. In this, you can kind of see the screen and the keyboard as like a 3rd tape, yeah. And that's gone in our BFF setup. Instead, we replace that with a 2nd head. So now this instruction that normally would output a byte from your data tape to your screen copies a byte from one place, from one head in your data tape to another head in your data tape.
You have two heads total?
Yes, in the BFF you have 2 heads. In the virtual one you have 1 head. Oh, wait, you have the instruction head as well, I guess. But I kind of wanted to not go too deep into the exact implementation details of BFF and give you more of a holistic overview of how it looks. So unfortunately, this is not valid BFF code. Pretend it is. So what we did in these first experiments was initialize a soup, which was just a huge array of random bytes, where each row in this array was 64 bytes and consisted of a set of valid instructions in BFF and just random bytes. I mean, so it was initially to be random. By chance, some of them are valid instructions in BFF, and the rest are not. What we then do is, at random, choose 2 of these tapes and pull them out and concatenate them together like this. Then we have now a 128-byte length tape, and we run this through our BFF interpreter. As before, this 1 tape is both your data tape and your instruction tape. So every now and then, the program might change some bytes in itself. It might overwrite or increment a byte or copy a byte or something. And once this is done, usually stopping by some maximum number of operations run, we take those 2 tapes and put them back into the soup. So the interesting thing was, if you repeat this process a lot of times, you get something like this.
Just a question.
Yes.
Is it a soup per cell? Or is the soup just all the cells?
At this point, we don't have cells.
Oh, okay.
Think of this as like just an array of
Okay.
set of rows of bytes, where each row is 1 tape. And you're just picking 2 tapes random, running them through the interpreter, putting them back. What this actually looks like, and this is a really nice visualization that Alex made, is something like this. So I'll just pause for a second. In this visualization, this is a soup. Each kind of column here is a very zoomed out view of your soup. Each row is again one tape. Highlighted green highlights are coding bytes in BFF, and non-highlighted characters are just normal non-coding bytes. On the right side, you have a very zoomed in view of, I think, this section of the tape right here, where you can see the actual bytes. Again, the coding bytes are highlighted and non-coding bytes are unhighlighted. And there's also some indications of where the heads are in each tape. And then if you do this process of taking 2 at a time, running them through BFF, putting them back, this is what the soup will look like as time evolves. If you look very closely, you can kind of see the number of coding bytes slowly increases, but very gradually. But then at one point, which we'll see in a second, so they definitely are modifying themselves at this point already. You see this phase transition, which is quite striking. And that's when 1 tape somewhere ended up forming a fully formed replicator that can, when these 2 are concatenated, copy itself from the first tape to the second tape. As soon as that exists now, because you're kind of putting them back in the soup and sampling them over and over again, your whole soup quickly gets filled with this replicator emerges. Called life emerging. So I guess people have a lot of like questions usually as soon as they see this. One of the most common questions people ask is kind of, is it crystallized to a single fixed point, or is like a replicator that works and just fills the soup and stays the same? And in the shorter time periods, not really, no. You see quite a lot of diversity emerge in these replicators. You see different forms of replicators. Some of them just copy themselves directly, some of them copy themselves in reverse. And they kind of continue evolving in our flux for quite a long time. I mean, my intuition for why this happens is because once the soup is filled with tapes, you might end up pairing a replicator with a replicator, and now the first replicator has to try and copy itself over the second replicator, and the second replicator has an interest to try and be, try not be copied over. So they're both competing and actually sometimes also collaborating. So we have seen like some interesting cases where replicators emerge that will use one of these for loops, one of these like square brackets, where the end of the loop is in the second tape. So it creates a loop that continues into the second tape, and the copying loop is through both these tapes. So it's quite interesting dynamics occurring there already.
Maybe I just don't understand something of the language, but what if you run something that doesn't halt?
So we have most things actually don't halt. We have a fixed limit on the number of operations that can be run across the tapes.
Yeah,
So just kill them after some time.
there's no hold basically in that
Yeah,
project.
there's actually no way, I guess, for
After like 1,000 steps, I think we just move on.
Yeah. I think even depending on the setup, sometimes we also let the instruction pointer loop around, like if we can reach it on the tape. But most of the time, I guess they almost always just get killed by.
There are some variants.
Max instructions.
If we go to the very end, then we halt, but it's not necessary. Like you can also just keep it like wrap around.
Yeah.
There must exist some soups that cannot lead to any replicator, right?
Yes, there are. Yeah, there. I guess for the setup I'm describing with BFF, not having enough randomness present is a condition that can prevent replicators emerging, and that can actually occur in two ways. You can either have mutations during runtime, or if you just seed the initial soup with enough randomness, if it's big enough and uniformly random, even if you turn off mutations during the actual runtime, you will still see replicators emerge.
When you say that replicator don't emerge, what's happening to the soup? I mean, does it settle?
Yeah, I mean, if you just, kind of a pathological case, it just
All non-coding bytes.
bytes or like very.
But I mean, when you say that there's not enough randomness, does it settle in a kind of static state, or does it keep
If I remember correctly, I think he just kind of
looking
stops.
chaotic?
I mean, it looks kind of chaotic. It depends if you initialize it somewhat randomly, but I think it just you don't see many modifications by code of code.
So it stabilizes.
Yeah.
I see.
I think I've seen a lot of failure cases where it felt there was like a self-replicator that was arising, and then something bad happens, and it's just chaos. And then until we kept looking, it was just chaos.
I see. But if it's always chaos and doesn't settle into a kind of a loop somehow, there must be a time scale where a replicator will emerge, right? Because you know that it exists, and if you don't settle into a loop, you must kind of
Yeah, I think
this
you would
self-replicator.
and have to end up in a loop. It might be a very big loop.
Okay, yeah.
Because everything's finite.
Yeah.
Exactly. Yeah, go ahead.
When you say that it doesn't immediately crystallize to a single replicator, how are the replicators different from each other? Is it just like random mutations in them, or?
Not even random mutations. Kind of how they mutation differs often. Like you'll see sometimes just this very prototypical replicator emerge, which is one that's in this video here. Okay, I can't say no. But sometimes you'll see ones that will copy themselves in reverse. You see ones that
I see.
sometimes ones that collaborate, as in they get paired up. The first one uses a loop that crosses the boundary of the two tapes to do the copying. So you kind of see different species of replicators emerge.
Is there like another phase transition after this, or is it kind of this is the phase transition that you guys have observed so far?
For BFF, this is kind of the main one we see, yeah. And then you do see some variation.
Sometimes we see self-replicators become more robust, so that they create like double loops for auto error correction.
Yeah, actually that's a cool one. So an interesting thing that does happen over time as well is if you have mutations on, I think only, they tend to get shorter over time. I think because shorter ones are more robust to mutations. And also, yeah, you get these kind of cool ones that have 2 loops. So if some mutation manages to exit the first loop, the second loop catches this kind of error condition and puts it back in the first loop, which is kind of crazy. So you see like kind of error correction occurring as well. Yeah, sorry.
Go ahead.
Did you, like you said, for a long time, how long, and have you reached an end to the flux of continuous evolution?
No, but we kind of do see it ending up in one particular replicator at the very end, not always the same one.
Okay, right.
And
But there's still flux, even if it's like the very end. So potentially, if you just ran it for longer, you could continue to.
Yeah, maybe. Yeah.
Yeah.
Don't have a great intuition around that. Yeah, and then in the paper, which maybe some of you saw, there was some work at looking at where bytes come from. So whether replicators emerge kind of as like a random sampling of programs, or if they are code modifying code. And for the most part, it's code modifying code. And there's kind of a nicer diagram in the paper itself. But there's some of the people I mentioned in the beginning are working on kind of a stronger version of this claim back in Zurich, trying to show that kind of symbiosis between even proto-replicators is required for replicators to emerge. But that's still not a production that works. And what might be interesting for this this talk is how do you measure replicators? So how do we check when they emerge without just looking at the soup? And oh, sorry.
Yeah, sorry. Just to make sure I understand. So there's 2 separate questions, I guess, about the nature of, say, self-replication. One is whether a program has a description of how to replicate, and the other is can it create the means to execute it? Because in this case, you're sort of operating externally on the strings to evolve them. There isn't really a sense in which they produce the means to execute, as you would need in a cellular automata where they have to create other crap, you know. But so here it's kind of about just this merging of it's almost like you have Turing, you know, it's not really Turing machines, but you have some model where you can kind of mix and match, and you're interested in the space of programs that are made by a certain mixing and matching, which can, you know, proliferate.
Yes, that's correct. Yeah. Yeah, the actual replication mechanism is sort of the physical rule of the world. Yeah. Well, sorry, the rules that the instructions it uses to replicate are the physical rules of the world. Yeah.
Do you know like autocatalysis in biology or like?
Mm-hmm.
So there's a lot of correlations there with like these soups that create pieces that together they create like networks that self-replicate.
Yeah.
We are not at the DNA level and polymerase level. It's more like autocatalysis right now.
Abstract.
Very abstract, yes.
Yeah. So what might be interesting is how we measure the replicators emerging. And it's actually quite a hard problem to measure for a number of reasons. You might think like, okay, I just check, has this program copied itself from the left tape to the right tape? But you see, I mean, there's a lot of like, kind of, not even edge cases. Most of them are like this. Sometimes they don't copy themselves perfectly. They copy just a part of the string. Sometimes they copy themselves a different offset into the second tape. So just matching the 2 tapes won't tell you if they copy themselves. There's also all these non-coding bytes that live alongside the replicators. Those sometimes get either modified or also copied when the replicator copies itself. And sometimes they copy themselves imperfectly. There's a lot of kind of these cases where it's very hard to actually define a reliable test for whether a replicator copy itself. So kind of the 2 paths we had to like measure this copying behavior was to try and generate heuristics for is this a copy, do some sort of fuzzy string matching or something, which some people now in Zurich are doing and using somewhat successfully. But at the time we tried something else, which was kind of initially to just measure the entropy of the entire soup and see how that changes. But the problem is that just pure random noise also has a very high entropy. We end up using this construction. So the point of this is to kind of ignore entropy over single bytes and measure entropy in the relation between bytes. And the way this looks is this: you just take the normal Shannon entropy of the soup over bytes. So that'll just be treating the distribution of bytes as their frequency in the soup of the soup size. And to kind of give a lower bound on, upper bound on column complexity, we use just an off-the-shelf text compressor. We actually use something called Broccoli, but I'll put zip here. And both of these will essentially have the same units, which is bits required to encode a byte in the soup. So then you could just subtract them, and this gives us a somewhat useful measure to look at the soup, because
You just concatenate the soup in whatever order it currently is.
yeah. So a perfectly uniform random string, both of these will in the limit of a long string, become the same value, which in this case log 2 of 1 over 256, which gives a value of 0. But a more interesting string like a replicator will be much larger than 0. So it kind of filters out these very, very basic pathological cases, but gives a nice indication of more complex and more interesting replicators. So what does it actually look like? If you look at that, if you go back to the same video I showed you earlier with the phase transition, if you take this measure and measure that on that soup, it looks like this. That's the green line. This complexity measure we have. So it starts out at 0, perfectly uniform random, and you have a small increase, which is interesting, of complexity before the phase transition occurs. And that's kind of bytes modifying bytes and kind of increasing the number of coding bytes, which we don't fully understand. That's kind of something we're still working on. And then the actual phase transition is here. This is an interesting case I'll talk a bit more about, which is there is a kind of virus that can emerge in these replicators, which kills them. We see it both in BFF and I'll show you later in ZFF. And here that emerged, kind of somehow the replicator fixed itself to avoid that being susceptible to this virus, and then did a full phase transition later. The other two lines here, unique, refer to, we call a token unique or we classify a token as a unique token when it's created either randomly in the beginning of the soup or through a random mutation. So in the beginning, you have a bunch of unique tokens. As time goes on, some of them get deleted, so the number of unique tokens in the soup kind of decreases. And then after the phase transition, you have some sort of balance between mutation rate, which introduces new unique tokens, and replicators replicating themselves.
What is the unique no transition? It's just a run where it happened to be no transition.
So that's what it looks like if you only had mutations and no actual execution of the EPFL programs.
Okay, I see.
We'll kind of stop like this.
Yeah, one addition is that one of the reasons why we stuck with this complexity measure is because it was very highly correlated with the unique tokens, which is more costly to keep track of. But the unique tokens might be more informative in the sense that it's telling you, actually now their soup is using always the same thing. And that suggests the replicators more strongly.
Yeah, so that was kind of a summary of the paper so far, and then a little bit in the paper also talked about Z80. We then did some experiments where we switched out the kind of the brainfuck interpreter, or the modified brainfuck interpreter, for a different instruction set from a real CPU called Z80, which predates me apparently, and also switched to a 2D soup where the tapes are concatenated with the neighbors. I'll show you how this actually looks in a second. So the instruction set we're using now is much larger. You can even have multi-byte instructions. So both in like complexity and explicitity, you can write much more complex functions. You can do memory copies, you can do more advanced loops, you can load things into registers in your CPU. And the actual soup setup looks like this now. This is again a beautiful visualization by Alex. So we have, this is zoom in. Each pixel here is one tape. I'll pause here.
So the tape is smaller, right? It's 16 instructions.
Yeah, so each square here, each pixel on the floor is one tape. It's just 4 by 4 bytes, 16 instructions.
The tape is still linear, you just put it
in. It's just wrapped here.
I see.
I think wrapped. Yeah. As a snake or scanline. And it's sort of like taking two tapes at random from the whole soup and pairing them. We always paired a tape randomly with one of its neighbors. That's what these arrows indicate. So this tape going here gets just concatenated with this tape here. And the whole thing gets fed through fed as a program to a Z80 interpreter.
Can the tape move, or they're stuck?
They're stuck. They also can't choose who they pair with, so they can't like intentionally move anywhere. Yeah, and then the smaller squares and some big squares are just representation of the instructions. We've manually colored a few of the bytes because they're actually, I will hand colored them, and when we're zoomed out, the color of one of these squares is kind of the average of the color of the bytes inside it, just to more easily visualize replicators. So this is actually going to be a replicator here. So we zoomed into the spot, and you can see the actual instruction pointer there kind of moving along. And as it gets paired with its neighbors, it keeps copying itself, and so on and so forth. An interesting thing here you'll see is that, I don't know if you caught that, but right there, the tape, it's so hard to see my mouse, copied itself. Let's see. And then disappears.
Yeah.
And that's, we'll see the effect of that in a second. That's kind of one of these viruses I was talking about, this corruption that occurs in these apes. And as you zoom out, you'll see it kind of take over the soup. But the interesting thing here is that instead of just becoming a fully bright green kind of filled area, you see that the tapes kind of die after they spread. So you have this wavefront of tape spreading. And that's, again, this.
How long does it take to make a simulation like this, computationally? Like this full simulation?
This is real time
in
a browser.
Nice.
Thanks to Alex Magic. It runs on, I use it, sorry, web GPU. Or WebGL?
No, it's like WebAssembly.
Oh yeah.
Okay.
Yeah, sorry.
WebAssembly.
So CPU then, yeah.
Four parallel workers on CPU.
Exactly.
It seems like these updates are synchronous, like you just pick a partition of two neighboring ones and you update them all at the same time.
In this simulation, yeah, exactly. Not all tapes get paired up, as you saw, but every kind of outer loop time step, some of them get paired up, get run through, and so on. Yeah. And that kind of covers everything we had in the preprint we put up. Nice to show you a few more kind of hand-picked examples of cool things that you can see in ZFF. So one of them is speciation. So here we have a soup a bit later in time with the same replicator we saw earlier, and we see a new replicator emerging here just by chance. I keep track of how many of them exist by just looking for these 4 bytes that constitute them.
Sorry, I might have missed this. Is there a random mutation, or is this just random recombination of what was already there?
I haven't traced exactly where it appears from. Like this,
Okay, but random mutation is enabled.
Yes.
Oh, okay.
Yeah. But you end up with a bunch of different species, and they're kind of competing at these wavefronts. You can see that for a long time, a lot of them exist or kind of coexist on this grid, and eventually, I think actually, maybe even a 5th type emerges. I can't remember. But you have this very nice kind of coexistence, competition between them, where they're really incentivized to, defend themselves against being replicated, or develop a better way to replicate.
So finally, in the right side,
with a new one here emerges.
Like using completely different principle for replication.
So these are kind of similar-ish instructions. I don't know why exactly one of them is better than other ones.
Yeah, I guess, I mean, you don't have to go into too much detail, but like, what is qualitatively different about these types of replicators? Is it something that you can explain easily, or is it like very specific to?
Not here right now. I do have this is all actually coming from an article we're putting out, where I do have like a debugger, and I step through exactly how a few of them work and why they copy themselves over, or why they kind of beat another replicator, for instance.
I see, okay.
I can quickly explain. So first, 3 replicators use the same mechanism of feeding like their body into one of like 2 8-bit registers and pushing it on stack. So they use the same replication mechanism. 4th is slightly different. Instead of putting immediate data, it uses indirect like lookup, to load data in registers. And somehow it has a bit different dynamics than the first three and seems to help them to replicate, like seems to be some symbiosis by clearing up this dead ground after that. And then the last one that appears on the right uses a completely different mechanism, discovers memcpi, which is like a special prefix instruction that can copy a chunk of memory from one location to another and uses this to replicate and eventually displaces everything. Actually, we saw a different mechanism, like in a different universe with different seed. There is another type of stack-based replicator that also eats everyone eventually, that swaps the content of register with top of the stack. There is a special instruction for that, and apparently somehow this is more efficient than just pushing itself instead. So they discover different features, which I wasn't aware of.
Do you guys think you've like exhausted? Yeah, I mean that's as for as little as I understand, that was satisfying. But is this something that you guys think you've exhausted, or like if you ran like 10 more seeds, you think you would still?
No, I
find
haven't
it?
exhausted.
Okay.
So, and then both in BFF and ZFF, we see this interesting kind of viral program, not even program, in this case just a single byte emerge, which actually kind of hijacks the copying mechanism of an existing replicator to copy itself, and then kills both tapes. So in this case, this is kind of a debugger I had. So these are two tapes that have been concatenated, tape 0, tape 1. Tape 0 is filled with a replicator of a very simple kind. This kind of green one we saw earlier. Tape 1 is filled with some junk and then has a single byte here at the end, which is going to become very important. As tape 0 tries to copy itself into tape 1, it kind of does so successfully, but doesn't copy itself over the last byte for off by one reason. And then actually ends up copying that one byte to itself. As soon as it does that, and the program counter loops around one more time, it actually then ends up copying that byte using its own kind of copy mechanism over both tapes. And this is what you're seeing when you saw that kind of replicator die early on and turn from green to black. It's this almost viral mechanism hijacking the copy mechanism to copy itself and then killing both tapes. And later type of replicators emerge that kind of reset this state to allow it to be copied over again by replicators. As it is now, it's kind of poisonous to any new one. So if a new replicator tries to copy itself over here, it also gets killed in the same fashion. So it's kind of a very, very interesting mechanism you see. And then here's a little glint, I would argue, of open-endedness. In this experiment, we took two replicators that we had, kind of early stage replicators that we found in two universes with the same conditions but different seeds. So you could as well have taken these from the same universe, probably with a very big grid, and found them in two different places. And we just placed them next to each other in a brand new, uniformly randomly initialized universe. As a kind of early replicators are pretty evenly matched, and they sort of have this front where they battle each other and they grow out through the rest of the soup, but neither of them kind of wins. They both expand backwards. And then right around here, we change the rate of mutations in the soup to increase it. Not by a crazy amount, but by a fair amount. And that ends up immediately killing one of the replicators on the left. Another one, for whatever reason, already is kind of resistant to that change, despite the two of them having kind of emerged under the same conditions. And I think that's a little bit of a hint towards that you can have kind of open-ended emerge in these universes, especially if you have a big enough grid. And why are these interesting? I mean, we've had a few talks today by kind of the linear folk as well, and linears that take the parameter with them. And I think those systems could probably kind of show the same behavior, given a big enough grid and a fast enough grid. This system I really like I really like the system because it can be made very, very fast. So Z80 is still being interpreted. You could take this and run the same experiment on kind of these bare metal assembly instructions on a laptop. And it's just very optimized for our current stack of hardware. You can just kind of scale this up as much as you wanted. So if you have some interesting behavior merge, it's really easy to kind of scale these experiments up and find even more interesting behavior. And because the environment of a given replicator is other replicators, they're constantly trying to kind of outcompete each other or even cooperate with each other. And I think that also is kind of a bias here towards creating more complexity or more open-endedness. And yeah, this isn't a very strongly backed claim, but my intuition is that this happens a lot faster than you'd expect if you're just trying to evolve kind of a self-replicating program by choosing it, as you would in a normal like evolutionary strategy approach. I think the same, my gut feeling is the same process that leads to these things emerging so fast probably could also be leveraged to do, for instance, learning at some point, or really kind of like a better version of evolutionary strategies. Here's another kind of imitation I had. Yeah, there's this old adage, right, about monkeys and typewriters that 1 million monkeys would actually never write for Shakespeare, even if you gave them a lot of time. And I think there's some calculation on Wikipedia that 10 to the 29 million monkeys of left the universe would have a trillion chance of writing Hamlet. But the truth of the matter is that in life, these monkeys kind of interacting, competing, knowledge sharing, predicting each other, after enough time actually end up doing that. And I think scaling these things up, you could do hopefully something similar in our FF languages. Yeah.
We have time for a few questions, and then we'll move to the next one. There was one right here.
I mean, I have a comment about the Hamlet thing. Like, if you sample one number between 0 and 10 to the power of 10 billion, it's gonna be a number. And then if you say, oh, what is the chance of sampling that number? It's 1 over all of them. But I mean, the significance of Hamlet is, you can't justify it like this. That it's just it could just be a random string. You know what I mean? Like,
I'm not quite following, sorry.
Like, let's say we choose a random number between 1 and 10 to the power of like 100 billion.
Yeah.
And it's a specific number. And then we ask ourselves, oh, what was the chance that we get this? And then you say like, okay, it's really amazing that we got this. It's kind of.
I don't think Hamlet was randomly sampled, right? Or
It can be a randomly sampled.
Yeah, but there's a kind
It's
of
not.
description length, right?
No, but I mean, we're talking about it after it's sampled. So
But
like,
a priori, you can still, I mean, even after you can say, well, there's only so many things I could have described with.
yeah, but
This
that's
works.
after all of these structures appear that give it meaning. So
yeah.
This-
That's kind of my point, I guess. That the same process of these structures of like what we find interesting or beautiful kind of emerged over time through these processes.
But I mean, you can't justify the significance of this post hoc after it emerged.
No, you could. I think you could, if you can argue that even a priori, there's not so many things you could have spoken about. It's like this random string, I think you could not have described it a priori. But the idea of, I think there's a way around this. I did suffer from this.
Like, what is the
I
question?
might try this. I'll think about it more.
Let's,
but, yeah.
let's,
There's a way
let's
around.
continue to another question because we don't have so much time, but we can discuss it later also.
This to me seems a bit analogous to, not in the sense of how it operates, but just in the sense of its complexity to like Lenya, in the sense that you have self-replicators that just don't conserve any quantity. And so it's hard to, I mean, of course, space is, in this case, your constraint, but
And time.
and time. But you can just fully replicate yourself. Have you considered using a language or just modifying Z80 so that there are no pure copy instructions? There's only like you can grab, you can move yourself, or like you can move an instruction. So say for a self-replicator, you'd have to just gather a bunch of junk instructions first, and then just move those junk instructions to the next cell.
Not that explicitly. Kind of in the same vein, one of the things I'm kind of trying to build right now is, or with a collaborator, is to let cells, or in this 2D case, cells choose which neighbors they want to match with. Because that would be quite interesting. That could A, lead to replicates being able to move across the grid, but also could lead to cellularity in the sense of cells being being these pixels here, because you can then have consistent matching of cells lead to like a bigger program emerging between 2 or 3 or 4 cells.
But there's some concurrent work that is doing chemistry with combinatorial logic, or combinators, sorry, and they can make it mass preserving. So like,
I think there's one by Tomasz Mikolov that was like artificial chemistry systems.
yeah, so like there's a direction that can be explored there as well.
I agree with the sentiment. I mean, life is kind of consumed by energy, and that decides a lot of how things occur. And something similar. I mean, there's some equivalence between like the space constraint and the time constraint.
Yeah. I mean, if you look like at a physical cell, it does have space constraint and time constraint, but it also just has mass constraint. It needs to acquire the mass before it splits in 2. So it's like sort of 2 out of 3, or maybe even there's a few more constraints out there. But
yeah,
still extremely cool.
Okay. Yeah. One last question maybe, and then.
So the program length is fixed now, right? Have you considered having it adaptively so it can also mutate length? You're bounded.
I haven't tried like the program itself being able to change its own length, but I'm hoping it's captured in this kind of choice of being able to choose your neighbor. Should effectively.
Okay, yeah, that's interesting.
Create chains of programs essentially.
Yeah, very cool.
Okay. I have another question, but let's talk later because I'm stressed about the time. So let's thank Eyvind again.